In the previous section, we took a look at the life sciences industry, and in particular, we looked at both the pharmaceutical and biotechnology segments. In this section of the course, we take a more detailed look at the process of drug discovery. The treatments we see out of these industries come in a wide variety of forms. The question is how to get there. How do we wind up with liquids, tablets, capsules, or whatever form the drugs take? We are going to use the graphic on the left of the slide as a guide to the process of drug discovery. The chart shows all of the stages that have to be gone through from the recognition of a disease or condition to the delivery of a treatment method to the public. We'll go through the steps in the graphic quickly and then go back and look at each element in more detail. The process begins with understanding the disease or condition that needs treatment. What are the symptoms, effects, how serious is it? Does it affect a small population, or is it more widespread and potentially profitable? The next step is the discovery phase, where we look for candidates to treat the condition. In the pharmaceutical industry, it might be small molecules, and in the biotech industry, they can range from genetic treatments to proteins to more elaborate approaches. Once we have found some possible candidates, we enter the preclinical phase. Here, we begin an evaluation of each of the candidates against the disease. It begins with the screening process to separate more promising candidates from those less likely to make it through the process. We begin with testing in test tubes and work our way through animal trials. In each step, the number of candidates is reduced. Once we are finished the first major series of steps, we enter the clinical trials phase of drug evaluation. The purpose of clinical trials is to evaluate the effects of the treatment in human beings. There are 3 critical phases in this process that review the safety of the treatment, its effectiveness, and patients' ability to tolerate the treatment and evaluate side effects. Once a treatment has successfully completed the clinical trials and information has been gathered through all stages of the research, the results of that work is submitted to the FDA for review. The purpose of the review is to determine whether or not the treatment is successful and suitable for putting into the market. In recent years, the clinical trials process has been extended to include post-marketing surveillance and comparative effectiveness against other treatments. These are often referred to as clinical trial phases 4 and 5. Their purpose is to make sure that the drugs are effective in a larger population and to test their effectiveness against other treatments so that physicians can recommend the best course of action for their patients. As you might expect, the process is a bit more complicated than that brief outline suggested. For example, the FDA oversees the process from the start of clinical trials through phases 4 and 5 during production. Before clinical trials can begin, a company must submit an investigational new drug application to the FDA. At this point, they want to show that they've done enough work to suggest a potentially successful treatment, and it is worth taking the next major step from animal studies to human trials. At the end of Phase 3, a new drug application is submitted to the FDA, which reviews all of the information gained during clinical trials, including safety, effectiveness, and potential side effects. At this point, as noted earlier, the FDA reviews material and determines whether or not the treatment is worth taking to the market. They also begin discussions about the type of packaging and labeling and any warnings that might be needed so that people can understand how to use the medications. The overall process can take from 10 to 15 years from the time the pre-discovery phase begins until a product is ready for market. Given that patent protection is only 20 years, this gives a manufacturer a very short time to generate income. The entire process can cost anywhere from $800 million to $1 billion per drug. During that time, anywhere from 5,000 to 10,000 compounds that start the process will be reduced to one. As we noted in the previous section, a successful drug can be very profitable. This chart shows a comparison of the profits and R&D costs for various drug manufacturers. For the larger or mature organizations that have well-honed processes, there is a considerable room for profit. But as you look to the right of the chart, you see smaller companies who were still in the R&D phase and have yet to release the first products. This chart is based on data that was available in 2002, so there have been significant changes between then and now. We looked at this chart in the previous section. We are repeating it here just to reiterate the level of profitability and income that can be generated by successful drugs. Now we will go and look at each one of the stages in the drug discovery process in more detail. The purpose of the pre-discovery phase is to identify and describe the disease or condition that we want to treat. We will also need to look at what the possible causes of it are. Is it bacterial or viral infection, a genetic disorder, a protein deficiency, or an overabundance of a particular protein? At this point, researchers will bring to bear a whole arsenal of diagnostic techniques to find and identify an organism or substance problem that may account for the issue. Once we've identified a disease, the question is how we treat it. If it's a bacterial infection, antibiotics might be appropriate. Vaccines can be used against several types of organisms, including viruses. Gene therapy might be tried as a way of alleviating the condition. Over a 3 to 6 year period, researchers will examine thousands of potential candidates for treatment and work their way down to perhaps 250 or so that look promising and are worth further investigation. Sometimes the discovery for treatment is anecdotal or observational. Aspirin was discovered when people realized that chewing on the bark of the willow tree could relieve some pain. Fleming discovered penicillin when he noticed that bacteria wouldn't grow close to a mold and found that the mold was releasing a compound that inhibited the growth and killed the bacteria. Much of the work in drug discovery is moving away from broad-spectrum treatments towards more targeted treatments and personalized medicine. Instead of looking for compounds that can treat millions of people, researchers are more focused on diseases and treatments and looking at ways that treatments can be tailored to specific individuals or groups. The problem is finding drug candidates. There are basically two issues that need to be addressed: identifying possible candidates and seeing what they might be effective against. The illustrations on the left of the screen represent catalogs from supply houses that contain many thousands of possible drug candidates. These are organic compounds that may or may not be effective against different diseases without damaging the patient. In addition to known chemical compounds, scientists are looking at a wide range of environments for possible drug candidates. These environments may contain unusual organisms that have unique chemical compounds. A frog's neurotoxin can become an anesthetic in dilute form. Chemicals used to protect marine invertebrates might be useful against bacteria or other disease-producing organisms. Basically, the search is looking for interesting organic compounds that may be useful in the treatment of disease. Once we find these compounds, then we need to figure out which ones are going to be effective against the problem researchers are addressing. How do you deal with studying hundreds of thousands of chemicals and their potential biological effects? One methodology used is called high-throughput screening. This is a large, sophisticated system that consists of both a large physical library of chemical compounds paired with an electronic database that describes compounds and the information researchers have gathered about them. The success of high-throughput screening techniques is due in part to standardization of the test platforms. In this case, they act like an array of many small test tubes. The importance of standards is going to be shown over and over again in this course as a critical factor in automation. The drawing on the screen shows the basic format of the test plates. Each circle represents a small test tube. Plates can contain from 96 to more than 9,000 wells. Their design allows them to be handled by robotic systems that contain components to add chemicals, heat them, wash them, and measure the reactions that take place. When screening tests are being performed, a researcher identifies the compounds that are to be used, and the robotic system gets the compounds and then puts them in the test bed and runs screening tests against them. The results are stored in the database that researchers can analyze and find useful indications of promising results. Care has to be taken making sure that the data is up to date so that expensive chemicals aren't wasted and to avoid unnecessary repeated testing. As a result, using high-throughput screening, the researcher can use the compound library within a highly automated system to select chemicals, test them against the targeted materials, and see the results with little human interaction. This improves the speed of evaluation and reduces the cost of the discovery process. Where high-throughput screening could be viewed as a brute force approach to finding drug candidates, molecular modeling is more of a designer's approach to the problem. As you can see in this video clip, molecules have shapes. They can bend, twist, and connect to themselves and other molecules in different ways. The molecules in our bodies undergo these twists and bends, and they can define how these molecules work. One of the ideas behind molecular modeling-based discovery is that we can try and find molecules that interfere with the behavior of a protein or a bacteria, and thus prevent the disease. Once we understand the shape of the molecule and where the binding site might be located, we can design molecules to fit the docking site and interfere with the progression of a disease. An easy way to understand what is happening is to consider a lock and key. Only one key will make the lock function. If we can fill in the keyhole, the key can't be inserted and the lock will not function. It's the same idea. If we block the active site on a protein, we can inhibit its ability to function. Understanding how proteins fold, which determines their shape, is a massive computational problem. One approach to dealing with it is to use distributed or grid computing. The idea is to take a large problem, break it down into small parts, and have those parts analyzed by computers over a very large network. The results are then recombined to complete the analysis. So far, we've only looked at two methods of discovering candidates for evaluation. There are others. One that is coming into play is the idea of data mining. Companies have been building up massive data files of chemical and disease interactions, and now they're trying to go back and look at that information to find out if there are possibilities hiding in previous research. This is one of the factors that is driving companies to converting their paper research notebooks into electronic form. Another approach to understanding disease is to look at the genetics of large populations. Companies and countries are collecting large numbers of genetic samples and personal histories in what is called biobanking. Freezers ranging in size from household chest freezers to warehouses hold many thousands of tissue samples that can be used for research purposes. They may be screened, looking for disease markers, and are matched with large databases of information about the samples. Each tissue sample is contained in a tube with a barcode. The barcodes can be used to identify the samples and serve as an index into the database. This type of research may allow us to identify predispositions to diseases and allow us to do a better job of tailoring treatments to individuals. However, there are significant issues that have to be addressed in doing this type of work. One is that we have to maintain the confidentiality of donor information. The database also has to keep track of sample locations, usage history, the amount of material on hand, references to all test results, and at the same time provide careful control over who has access to the samples and data, so the material is used properly and not wasted. While this is a large database problem, the solutions to managing it range from adding features to existing laboratory information management systems to collaborative projects to build open-source biobanking management systems. The discovery phase is all about finding candidates as potential treatments. Once we've found the candidates to work with, we begin the preclinical phases. This work is first done in cell cultures and then progresses to animal studies. Often, when you hear news reports about certain compounds being effective against diabetes, cancer, or some other disease in mice, and that treatment will not be available for human use for several years, the statements are based on work done in these preclinical phases. That's because they're still in the early phases of study and have yet to enter human clinical trials. In preclinical trials, researchers are trying to determine how safe and effective a drug is, and whether or not there are any undesirable side effects. They're also trying to understand what happens to the drug in the animal's body to better understand how it operates. They may be interested, for example, to find out if the compound leaves the body too quickly or accumulates in certain tissues that could cause problems. They also want to find out if it is carcinogenic or whether or not it causes the development of birth defects. In addition, preclinical researchers need to determine what kind of formulations should be used to deliver the drug and how stable those formulations are. This is where they determine whether a compound should be delivered as a pill, capsule, liquid, and what the dosage strength might be. The stability information will give them an idea of what the formulation's shelf life will be. The mice and cell lines that are used in this phase of testing are usually clones. It is essential that databases be maintained to keep track of the animals, where they came from, when they were obtained, and their histories. This information will be used to support investigative drug applications and to show that the companies are meeting regulatory guidelines. The reason clones are being used is to have a better understanding of the effects of the drugs on the animals and eliminate the variability that the animals themselves can introduce. Before cloning, testing on animals could yield data that looks like the chart on the left. Although a trend line can be drawn, it is hard to justify given the scatter in the data before clones were introduced. Once testing began on cloned animals, the variability tightened up and the trend lines became more obvious. Researchers have a complex job of identifying diseases, understanding the causes, and identify possible candidates for evaluation as possible treatments. While research labs have been looking at potential candidates, testing laboratories have been busy carrying their portion of the work. Among the tasks they've been performing are things such as structure determinations. Often researchers will be working with chemicals and compounds extracted from biological sources, and they may not know what the structures of the active ingredients are. One of the tasks that the testing laboratories perform is determining those structures, so the researchers will know the precise chemical composition of the materials they are working with. In addition, once they know what the structures are, they have to determine test procedures that can be used to evaluate the purity of the materials, for both research and manufacturing purposes. These test procedures are also used to determine and verify the dosages and formulations used, and to evaluate the drugs as part of product stability evaluations. Information technology plays an increasingly important role in the drug discovery process, as more and more of the work moves from manual, hands-on efforts to the use of electronic systems. The product components that are listed on the screen are just some of the elements that are needed to support both research and testing laboratories. It isn't simply a matter of purchasing products, it's a matter of purchasing and implementing the right products, and meeting the often elusive goal of integrating them into functioning systems. Where once the goal was simply to put task-oriented systems in place, we are now looking at tying things together to improve efficiency and reduce costs. Once researchers have completed the preclinical phase and identified the compounds that they want to go off and study in human subjects, we move from discovery and preclinical work to clinical trials. We've moved in stages from looking at thousands of compounds to hundreds, to very few possibilities. Before we can do this, we have to pass a significant hurdle. That is the review and approval of an investigational new drug application by the FDA. Once this is done, we can move to the next phase of the work. This is a significant step for a company and often results in press releases, and if they are a publicly traded company, a jump in the stock price in anticipation of improved market capitalization for the company. The purpose of the clinical trials is to evaluate the effectiveness of the treatment, look for any undesirable side effects, and determine the suitability of the treatment in the marketplace. The overall time frame can range from 6 to 7 years, with an increasing number of volunteers as we move through the various phases. Work starts with a small number of patients to determine safety, and then once that's established, moves to populations of thousands of individuals. The processes in starting the clinical trials involves first finding volunteers for each one of the phases, keeping track of those individuals, and managing the program. The management includes records of dosages, the results, side effects, and reporting. This is a massive and expensive data collection and management process. We'll cover the technologies used in more detail later. One of the problems with narrowing the range of candidates down to a handful or even a dozen is that it still leaves you facing a very expensive research path. In 2006, the FDA acknowledged an intermediate step called Phase 0 to help companies further refine the candidates they wanted to work with. Phase 0 provides a company with the ability to do some small-sized population human trials to get an initial read on the potential behavior of drug candidates in the human body. These studies do not provide any data on safety because the dosages are generally too small. The primary purpose is to help companies understand what direction they want to take and rank the candidates in order of priority. There is some question about the effectiveness of the Phase 0 approach. In clinical trials, Phases 1, 2, and 3 provide the classical study path. The work in the trials is performed on volunteers, and it may be contracted to outside organizations that have more experience and support systems for working clinical trials. The primary responsibility for the conduct of the trials still lies with the contracting organization. As with any regulatory activity, a company can hand off its responsibility to someone else. Clinical trials may involve more than one drug and can be halted at any time if adverse effects are determined. The typical clinical trials process begins in Phase 1. This is done on a small number of individuals, and its primary purpose is to determine the safety of the drug or treatment before moving on to large-scale trials on human populations. During Phase 1, researchers will be looking at a human's ability to tolerate the treatment, what happens to the chemical compounds that make up the drug in the human body, and what the biochemical and physiological effects are. It's essential that the safety of a compound be investigated before it's used on hundreds or thousands of other humans. As a result, this step is a critical step in the process. When we reach Phase 2, the initial concerns about drug safety have been answered, and researchers can be confident about moving on to larger populations of volunteers. With populations in the low hundreds, we can begin to seriously evaluate the effectiveness of the drug and continue to evaluate safety issues. In addition, researchers can evaluate the effectiveness of the treatment at different dosage levels and find out whether safety or effectiveness varies significantly with dosage. It's important to find out what the minimum effective dosage is to avoid overdosing patients and developing side effects. In Phase 3, we move on to larger populations. Notice that the phase process is a very cautious progression, moving from a small number of people to larger and larger populations as the safety and effectiveness of the treatment are demonstrated. In these larger populations, we continue to evaluate safety and effectiveness in what has now become statistically significant populations. This means the test results that were initially performed on small populations and might be subject to question because of the population size have now moved to populations that are big enough to clearly demonstrate the results of the potential treatment on patients and clearly show any safety concerns and potential side effects. The work at this level will also determine labeling restrictions and potential interactions with other drugs. We frequently see advertisements for drugs in magazines and on television containing statements about restrictions on who should be receiving treatment, potential side effects, and when to avoid taking these drugs in combination with other treatments. These labeling restrictions are developed in this phase of work. The results of Phase 3 in particular will be used as the basis of submissions to the FDA and having a product cleared for release to the market. As you might imagine, the amount of data that has to be taken care of for thousands of individuals is enormous. Information technology plays a major role in collecting, organizing, and allowing researchers to work with the results of the clinical trial process. The 5 items you see on the screen play a major role in conducting clinical trials. There is a rapid shift from manually collected data to electronic data entry to smooth the process. We'll take a look at what's in these packages and what they do in the next few slides. Clinical trial management systems are the final repository for all the data collected in the various phases of the program. This is, in effect, a large project management system that contains all information for planning, preparation, reporting, and managing the clinical trials process. All data collected, interviews, contact information, and so on is ultimately placed in the software. Because of the number of people that participate in the process from a wide range of geographies, systems have to be available 24 hours a day, 7 days a week. If a contract research organization, a CRO, is participating in the study, the software becomes more critical because of the need to get information from the CRO and coordinate it with the primary company's research departments. The initial data collection is done through a clinical data management system. Data can come in from a variety of forms, including paper-based reporting, which is gradually being replaced by web-based systems and voice response systems. Electronic data capture sounds a lot more impressive than it really is. Basically, it's a conversion of paper records to electronic format, where attention has to be paid to the accuracy and validity of the data entered. Typical software consists of form-based systems with provision for duplicate data entry so that the entered information can be verified, errors detected and corrected, and reports generated. Because these systems are labor-intensive, requiring both forms to be filled out on paper and then transferred to electronic systems, more cost-effective methods are being used to replace them. Two of the technologies currently being used consist of interactive voice response systems, which prompt the patient volunteer to answer questions and provide descriptions, which are interpreted by speech recognition systems or manually transcribed. With the wide availability of web-based systems, people can log into a website and enter the data needed as the study progresses. Aside from the need for the patient to verify the data, all data movement is electronic and as a result, much more efficient. Information technology systems provide a mechanism for collecting data in a variety of formats, depending on the capabilities that are available to the patient volunteers, and moving it through data management systems and ultimately into the clinical trials management system. Smaller companies or those new to the clinical trials process may determine that it makes more sense to outsource the work rather than take it on themselves. This way, they can spend their time continuing research efforts and leave conducting the trials to companies that are more experienced in these phases of the work. There are two types of organizations that can be used for outsourcing. The first are the organizations that actually do the clinical trial management. These have been referred to as clinical research organizations or contract research organizations. The second are contract manufacturing organizations that fill a significant gap for the clients. In order to conduct a clinical trial, you need materials, the drugs that are going to be tested, in large enough quantities to support the clinical trials themselves. Producing the material is often left to contract manufacturing organizations that have the skills and equipment to produce the necessary quantities of material without requiring the client company to build the manufacturing process themselves. Clinical research organizations have the experience and background and systems to recruit patients, take care of monitoring and data management, provide biostatistic services, and provide regulatory oversight for the entire program. For a small company or one new to the process, it makes a lot more sense to deal with an experienced research organization than it does to provide the staffing, hardware, and software systems and climb the learning curve of managing a clinical trials program. Contract manufacturing organizations have the experience needed to produce quantities of material in the dosage form that researchers need. They understand how to scale up benchtop processes to small-scale manufacturing operations. They can also provide quality control testing needed to ensure product purity and dosage uniformity. In short, they act as manufacturing organization without requiring the client company to build one. They can also provide all the regulatory oversight necessary to meet the FDA requirements. Contract manufacturing organizations can fill a significant gap between the research process and full-scale manufacturing and production facilities. As a result, they provide a very useful service. They also eliminate the need for the client company to invest in a construction program before they have a viable product. There is a lot of competition for clinical trials work, both domestically and internationally. India and China, for example, with their large populations, are actively seeking this type of work. There are concerns, however, with the quality of the data and the integrity of the results. There are also ethical concerns when working with companies in countries where legal issues can be compromised. From the information technology standpoint, there are issues of systems compatibility that have to be addressed in order to ensure that data can move from the contracting organization databases to the client company's data systems. The ethical issues are not trivial. The report noted on the screen, for example, shows a posting which states that a contract organization faked all the data that was returned to the client company. The full report states that not only this client company, but several others were required to completely rerun their clinical trials program because of questions about the original data coming from the CRO. When everything is done and a company is satisfied that it has a viable candidate for treatment of a disease or condition, it then makes a new drug application, usually referred to as an NDA, to the Food and Drug Administration. This is a culmination of years of research and development, evaluation, clinical trials, and data review and analysis. The NDA tells the FDA everything they need to know about what the drug is, how it was tested, the benefits of its use, final formulation, and how it will be produced and distributed. At this point, it is up to the FDA to review the material and make judgments about its suitability for market. There may be questions and answers going back and forth, talking about labeling and other issues, but ultimately this is the final hurdle that has to be crossed before a drug or treatment can go to marketplace. Information technology plays an important role here. This is the point where everything comes together. All the research work and so on are pulled together into one massive set of documents. The reports and all the supporting data are submitted to the FDA on electronic media, replacing the boxes of paper that would have been required otherwise. All this data has to be maintained and backed up and archived by the information technology groups. The data represents a very large expenditure of money and needs to be adequately protected. In addition, the data has to be accessible to answer any questions the FDA might have. That means it has to be electronically organized so it can be accessed and worked with in a timely fashion. Once the NDA has been approved, the company can then move forward and begin marketing the drug and finally generating some income from years of research and development work. The review process, however, does continue. In the past few years, 2 new phases have been added to the clinical trials process, both of which occur after the product has gone to market. In Phase 4, the company continues to monitor the long-term effectiveness and impact on patients in case there are conditions or situations that occur that will only show up after long-term use. They also monitor the cost-effectiveness of the treatment in comparison to other treatments to make sure that patients and doctors are getting the best information and making the best decisions on treatment programs. In addition, the drug may be used in populations or for conditions that were not considered as part of the original study. In Phase 5, we see the development of marketing surveillance studies and trials to determine whether or not products are being properly marketed and presented to the population. Advertising is reviewed, the effectiveness of drugs is continually reviewed and evaluated against other candidates. There is considerable overlap with Phase 4. The material provided in this module is an overview of the drug discovery process. In reality, there is a lot more depth and complexity to the work than what we have discussed so far. Our intent was to provide a context for understanding laboratory work and the role that information technology plays. This is also an area that is continually changing and developing with new regulations and new technologies and approaches replacing older ones. If you want to keep yourself aware of what is going on in the field, publications such as the ones on the screen are good resources for understanding the field as it develops. Life Science Leader magazine contains articles about regulatory issues, information technology issues, clinical trials, and other areas where information technology plays an important role.